So today's discussion is about receptor and intracellular signaling. It's a very high yield topic for USMLE exams. No matter what exam you are going to take, this topic is very important. Now, let's say that it's about how our cell works internally. That means how the receptor binds and what are the changes that happens inside the cell. So as you can see that receptors and ligands. So what is receptor and what is ligand? The receptor is any macromolecule where this ligand binds, and due to the binding of the ligand, there is some biological activity or change. All right. Okay. So the receptors are macromolecules where the ligand binds, and due to the binding of the ligand to the receptor, there is some biological change. Now, as you see, that examples of ligands. The ligands can be hormones, different hormones in our body. They have their particular receptors. Then any neurotransmitters, drugs, these are examples of receptors. So the main core thing is that these ligands will bind to the receptor, and due to the binding of the ligand to its receptor, there will be some biological change. Now, largely, these receptors are divided into two types. One is the cell surface receptor, another is the intracellular receptor. You can understand that some of the ligands, they cannot go through the cell, right? Because some of the ligands have some properties. Like if I say a ligand is a peptide hormone, now this protein thing may not pass through the cell membrane. Maybe that ligand is a large charged molecule that may not pass through the cell membrane. Now, when they try to bind to their receptor, they will want their receptor to be in the cell surface. That means if I draw a cell here, these receptors, some of the receptors will be in the cell surface, embedded in the cell surface, and some of the receptors may be inside the cell. Now, these receptors, you can understand, those receptors in the cell surface, therefore the receptors for some protein molecules or some large charged molecules which cannot pass through the cell membrane, lipid, and we know that in the cell membrane there is lipid. Now, some of the receptors are inside. What does it mean? These receptors are for molecules which can pass through this membrane, and they can bind internally into their receptor. So what type of molecule can pass through the cell membrane? Either this molecule is very small, or either this molecule is lipophilic. That means they're lipid soluble. They can pass through the lipid and they can bind to their intracellular receptor. So largely, receptors are two types: cell surface receptor and intracellular receptor. Next thing is the cell surface receptor. These cell surface receptors are divided into the several classes. Like first, we see that two areas we can see. One is the extracellular space, another is the cytosol, the cytoplasm. And as you can see in this is the cell membrane, right? And here, four types of receptor we can see. One is the ion channel receptor. The name implies itself, or name says about it, that when there is some ligand binding to these ion channels, some molecules, some ions can pass through it. That means here you can see this thing, this pink ball. It's the ligand. It binds to its receptor, and when it binds here, these ions are passing through this cell membrane. Next, you see the 7-helix transmembrane receptor, or 7-pass receptor. This receptor is also called the serpentine receptor because they look like snake, right? And these receptors, as you can see, they all have a extracellular domain, and there is an intracellular domain, and they are passing through the cell membrane 7 times. All right. And most of the G protein coupled receptors that we discuss are the 7-pass or 7-segment receptors. Next is the receptor that also passes through the cell membrane, but they just pass it one time, and they have some enzymatic activities inside. Now, based on the enzymatic activity of this receptor, we call it receptor with intrinsic enzymatic activity. That means this receptor itself has an enzymatic activity or enzyme containing domain inside. And the other receptor, as you can see here, this receptor mediates its biologic action through some enzyme, but it does not have its own enzyme. Right? Now, when we discuss it, sometimes maybe you know that sometimes we say that some receptors are tyrosine kinase receptor, and some receptors are non-tyrosine kinase receptor. The tyrosine kinase receptors are those receptors which has its internal tyrosine kinase activity. What is kinase? Kinase is an enzyme, and whenever you see the word kinase, it means that enzyme can add some phosphate group to its substrate. That means if I say tyrosine kinase, it means that some enzyme that adds a phosphate group to the tyrosine residues. What is tyrosine? Tyrosine is an amino acid. Now, some of the receptor, they have intrinsic tyrosine kinase activity. That means they have their own tyrosine kinase inside them, or own tyrosine kinase activity that is a particular property of them. But some of the receptor, they don't have this intrinsic tyrosine kinase activity, but still they have some enzymatic activity, and they run those enzymatic activity with the help of some other enzymes. That means in this case, number C, you see that this is the tyrosine kinase receptor, or receptor with intrinsic enzymatic activity. That means it has its own enzymatic activity. This internal part of the receptor, it has its own enzymatic activity. But this receptor, it does not have this internal enzymatic activity, and it takes helps from other enzymes to do that intrinsic enzymatic activity. All right. Okay. Now let's see that what else is here. Okay, so these are the names of the receptors. Now here is the first thing that we discuss is the GProtein coupled receptor, very common in our discussions of pharmacology and physiology. We discuss G-protein coupled receptor many times, right? Now, how this G-protein coupled receptor works. Now, this G-protein, these are some 7-pass protein. That means that passes through the membrane 7 times. And this G-protein has several types. Like some of the G-protein are stimulatory G-protein. Some of the G proteins are inhibitory G protein, and some of the G proteins have some other activity. We call it GQ protein. There are a lot of other G proteins, but for the USMLE step 1 purpose, we discuss these types. First, you see the GS protein coupled receptor. That means the receptor which are bound to the GS protein or G stimulatory protein. Now, this G protein or GS protein coupled receptor, this name, what does it mean? It means the receptor has a function internally. Receptor has a function internally. Those internal protein that it does the function with, those proteins are called G protein. That's why they're called G protein coupled receptor. Now, as you can see, that this ligand is binding here in that receptor. And this receptor, whatever the receptor is, this receptor is bound to this G protein. This collection of protein is the G protein. Now, as you can see, that this protein has three component. One is the alpha, one is the beta, one is the gamma component. 3 components of that G protein. Right? Now, this G protein, usually, it remains bound by GDP. This G protein, usually in the alpha component, it remains bound by GDP, right? That's why maybe they're called G protein, because they are bound to the guanosine diphosphate. Maybe that's why they are called G protein. Now, this GDP bound G protein, it's usually inactive. But whenever the ligand binds here to the receptor, whenever the ligand binds here to the receptor, there's some signaling that goes to that G protein, some signal it receives. And what it does, it attracts the GTP. The GTP is circulating in the cell. Whenever this ligand binds here in that receptor, then this G protein has an attraction towards the GTP. Now GTP comes and GDP is removed. So whenever GTP comes here, GDP is removed. And not only GDP is removed, this whole alpha subunit will be detached from the beta and gamma subunit. So again, to recap how the GS coupled receptor works. Now first, as you see, that ligand binds to the receptor. And when the receptor is the G protein coupled receptor, and this is your G protein, this trimeric protein is your G protein. Now this G protein has three subunits, alpha, beta, and gamma. The alpha subunit has GDP attached to it, but whenever this ligand binds to this receptor, this alpha subunit has a signal, receives the signal, and it attracts the GTP and it gets rid of the GDP, right? And now also when this GTP binds to the alpha subunit, as you can see, alpha is detached from the beta and the gamma receptor. Now when this alpha is detached, this GTP bound alpha, where it goes? You might notice that there's a small s here. This s denotes the G stimulatory type or stimulatory type of G protein, GS protein. That's why they wrote that small s here. Now, maybe it is a GI protein, then you would have find GI. A small I is written here. If it is a GQ protein, small q written here. Now, mostly they work like in the similar fashion initially. Now, this GTP that is bound to the alpha subunit of the GS protein, it stimulates some enzyme that stimulate the enzyme. This enzyme name is adenylyl cyclase. Now this adenylyl cyclase will convert the ATP into CAMP. Okay. When this GTP, this bound to the alpha subunit, it stimulates this enzyme adenylyl cyclase, it will convert the ATP into CAMP. What is CAMP? Cyclic adenosine monophosphate. Now that CAMP, this cyclic adenosine monophosphate, it will work on certain protein. This protein, as you can see, is a inactive PKA. What is PKA? Protein kinase A. Protein kinase A. These proteins are having several enzymatic activity to run the intracellular functioning inside the cell. Now this inactive PKA, with the help of CAMP, it becomes active protein kinase A. So when this CAMP binds to this inactive protein kinase A, it becomes active protein kinase A. And as you can see, these CAMP molecules are actually removing this R group. As these R groups are removed from this protein becomes activated. And when this protein becomes activated, it goes and it works on other proteins to run the biologic action. Maybe it starts to change some enzyme activity. Some enzyme will be activated. This target protein can be several things. Depends where this cell is working. Maybe some enzyme activity increases. Maybe some ion channel is activated. Whatever it is, that's how your GS protein works. So again, to recap, first, the ligand binds to the receptor, the GS coupled receptor, then it leads to the binding of GTP to the alpha subunit. And whenever GTP binds to the alpha subunit, this beta and gamma receptor, they are detached. Sorry, beta and gamma subunit are detached, and this alpha subunit along with this GTP binds and activates the adenylyl cyclase that leads to conversion of ATP to CAMP, and this CAMP activates the inactive protein kinase A to active protein kinase A, and that runs the subsequent functioning or subsequent uh, functions or signals in the cell. Now, this thing, as you can see, that this beta and gamma subunit, when they are detached from their alpha subunit, they have a tendency to inhibit this ligand receptor action. That means they were here together with alpha. Now, whenever this signal came, this alpha changed its mind and it is detached. Now, this beta and gamma subunit, they want to inhibit this action of the ligand and receptor interaction. How they do it? By some other protein, GRK, and they are phosphorylating that receptor. And there is another complex protein that comes here that is beta-arrestin that helps to inhibit the action of this ligand receptor interaction. Now, when this ligand receptor interaction is inhibited, the signal is lost. Then what happens? This beta and gamma has a chance to bind to the alpha subunit again. But the thing is, when alpha has GTP attached to it, it does not bind to the beta and gamma subunit. But when alpha has GDP only, then only it binds to that beta and alpha subunit. Now, this binding, this conversion of GTP to GDP, it needs to be happened, right? So what happens that this alpha has its intrinsic activity, intrinsic enzymatic activity, through which it converts the GTP into GDP. So alpha has its some intrinsic complex activity through which this alpha, whenever it binds to the adenylyl cyclase and it activates it, after that, as you can see, that this alpha subunit bound to the GTP converts into GDP containing alpha. Now, one thing you may say that, okay, whenever this alpha is stimulating the adenylyl cyclase, maybe it is giving off its energy, and that's why maybe it is converting into this GDP containing alpha. That can be one possibility. But another thing is that there's some action in this alpha, there is some intrinsic activity in this alpha that converts this GDP into GDP. And whenever this GDP bound alpha is available, that again binds to this beta and gamma subunit. And again, they go and sit here with the vicinity or with this affinity of this receptor. Then again, when the ligand comes, another signal, alpha again detaches and then run all of this action. That's how your GS coupled receptor works. Now, as you can see in this picture, that it is showing the action of the protein kinase A. Here in this picture, we saw that this protein kinase A works on the target protein, but we don't know what are the target proteins. I gave you some example that this target protein may be some protein activity, some kinase activity, who knows? It increases those protein activities, any enzyme activity or any other protein in the body. Now here in this picture, in the next picture, you can see that this protein kinase A is working in the calcium channel so that the calcium is entering the cell and this calcium is running some action in this body. Now here you see both the GS coupled function and GI coupled function shown together. The GS protein, the GS coupled receptor, as you see there, beta 1 and beta 2 receptor. Now this the ligand for those receptors is norepinephrine and epinephrine, we know it. And when this GS coupled receptor is stimulated, this G protein will act like the same that I just discussed. That means alpha will be detached, the beta and gamma will be detached. This alpha bound to the GTP will activate the adenylyl cyclase. Adenylyl cyclase will convert the ATP to CAMP. This CAMP stimulates the protein kinase A to be active, and this protein kinase A stimulates these calcium channels. And when these calcium channels are activated, the calcium enters the cell, and this calcium, by entering the cell, it stimulates the smooth reticulum or smooth endoplasmic reticulum, which are called sarcoplasmic reticulum, not smooth. I'm sorry for that. SR is sarcoplasmic reticulum. Sarcoplasmic reticulum. Now, the sarcoplasmic reticulum contains calcium. This is kind of a bag of calcium, and through which, when this calcium stimulates this SR, there is calcium-induced calcium release here, and this calcium can lead to contraction. But not only it does the contraction, maybe it's a muscle cell, that's why they're talking about contraction, but it can also lead to increased enzymatic activity, because we know that calcium is needed for increased enzymatic activity or activation of enzyme. Now that's how your that's how your GS coupled receptor works. Now how the GI coupled receptor works, that means how the G inhibitory type of protein receptor works. Now acetylcholine and adenosine, this thing binds with the receptor, and when they bind with the receptor, they stimulate the GI coupled protein here, the GI subunit of the G protein. And this thing, as you can see, it inhibits the adenylyl cyclase. Now, the same thing will happen here, that there is a alpha, there is a beta, there is a gamma. This alpha subunit will inhibit now the adenylyl cyclase. In case of GS coupled receptor, we saw that the alpha subunit was stimulating the adenylyl cyclase. But in this case, the GI coupled receptor is inhibiting the adenylyl cyclase. And that thing leads to every other action that is happening here will be inhibited. Because you can see that if this step is inhibited, the subsequent steps will also be inhibited. And that thing will lead to inhibition of this intracellular functioning. That's how your GI coupled receptor also works. So in a nutshell, that GS couple stimulates the cell to be in a positive way, the GI in a negative way. But both of them are GI coupled, G protein coupled receptors, and this G protein works by the alpha subunit when it bounds to the GTP. That's it. Next thing, as you can see here, that what happens to the CAMP? That thing you need to know, that what happens to that CAMP that we got from this ATP conversion, because as you can see that this ATP converts into CAMP by this enzyme adenylyl cyclase. Now this CAMP, if it presents inside the cell, if it is present inside the cell, what it can do? It can run these cascades of events, right? As you can see that the CMP can activate proteins, and there is a lot of cascades of events that runs here. Now, that if this CMP stays in the cell forever, then there will be this signaling forever. Now, nothing is forever. There is stoppage of everything. Now, that's how the CMP will also be removed. And this enzyme, this phosphodiesterase, is the enzyme that cleaves the CMP again to AMP. Now, when the CMP converts into AMP, it is not CMP anymore. And when it is not CMP anymore, it does not work, or it stops functioning. Now, everything needs to be stopped, and it's the intrinsic way of the cell how they stop the CMP from functioning. All right, that is your GS and GI coupled receptor. Now, next is the GQ coupled receptor. Now, here you see that GQ coupled receptor, again, a G protein coupled receptor, and this G protein coupled receptor, again, almost the same thing. Like here is the alpha, the beta, the gamma subunit. Here is the receptor, and here is the binding of the ligand to that receptor. Now, when it binds here, as you see that GDP is bound earlier, but whenever this ligand binds here, the GDP removes or GDP is lost, GTP comes here and binds here. Now this GTP, it detaches the alpha subunit from the beta and gamma subunit. Now this GTP bound alpha subunit for this GQ protein, we are discussing the GQ protein, GQ coupled receptor. Now this GQ alpha, or the alpha subunit of the GQ protein, it now stimulates one enzyme in the cell that is called the phospholipase C. Now, this phospholipase C, what it does, it activates and it works on some other component of the cell. Hold for a minute. All right, sorry for that. So here you see that this G phospholipase C, this phospholipase C works on the another component of the cell membrane that is called a PIP2. That means phosphorinositol diphosphate. Now, when it works on this PIP2, it converts the PIP2 into two components. One is the diacylglycerol, and another is this IP3, diacylglycerol and IP3. Now, this diacylglycerol and IP3 has different activities. Like our diacylglycerol, it works on this protein kinase C. It works on the protein kinase C. And remember, in our GS protein, we found protein kinase A. Now, this is protein kinase C. Now, this protein kinase C activated, it becomes activate, and it does the almost same function like the protein kinase A. That means it will work on some protein. That protein will convert in its shape, and it will change its structure. It will be activated, and there will be certain action in the cell. Here, as you can see, that this activity of the protein kinase C leads to ultimately decreased calcium current in the cell. But and on the other hand, you see that this IP3, it stimulates the endoplasmic reticulum containing the calcium ion. These calcium ions are released, and they also activate this protein kinase C. At the same time, this calcium activates other enzymes. So the basics here, what you need to know that how these proteins are working, that when this GQ coupled receptor is working, then there will be activity of, uh, like this, that there's a G protein GQ coupled trimers, that means alpha, beta, and gamma subunit. um, this alpha subunit will be detached with GTP, and this GTP containing alpha subunit will stimulate the phospholipase C that will stimulate, or that will work on the PIP2 to convert it into IP3 and diacylglycerol. And this diacylglycerol works on the protein kinase C, so that this protein kinase C will activate or work on several proteins. Now, this IP3 works on the endoplasmic reticulum, and that releases the calcium, and this calcium works on other enzyme activities. That's how your GQ coupled receptor works. Now, here's one example of this GQ coupled receptor. As you see, that alpha 1 adrenergic receptor. This alpha 1 adrenergic receptor, also you can see angiotensin receptor. I'm not sure what is that. The vasopressin receptor, some muscarinic receptor. They have this GQ coupled receptor activity. Right now, this GQ coupled receptor, whenever these ligands bind to this GQ coupled receptor, as you see, they are activating the phospholipase C. That phospholipase C works on the PIP2 of the cell membrane, that means phosphoinositol diphosphate, and it converts into inositol triphosphate and DAG. This inositol triphosphate works on the calcium release from the sarcoplasmic reticulum or modified endoplasmic reticulum. By the way, sarcoplasmic reticulum is then another name for modified endoplasmic reticulum. Now, this calcium do its job, and this DAG, this diacylglycerol, stimulates the protein kinase C to do its job. That's how this GQ coupled receptor works. So here's a list that how this autonomic receptor are divided according to their G protein activity, and what are the activities that is given very uh, briefly here. So don't worry, I'll send you this notes after this session. So if you want to go over it again, you can go over it. Next thing is the that's our G protein receptors, and mostly the serpentine receptors. Now here you see the GABA receptors. The GABA receptor GABA stands for the gamma amino butyric acid. We know that GABA is an inhibitory substance, inhibitory neurotransmitter of the body, right? Now this GABA receptor, as you see, that it has two types of receptor. One GABA receptor is GABA-A receptor. Another GABA receptor is GABA-B receptor. Now, GABA-A receptor, this is not a 7-pass receptor. This is just a single pass receptor, and it's a ion channel receptor. That means it works as the ion channel, GABA-A receptor, right? But the GABA-B receptor, this thing is a 7-pass receptor. As you can see, that these GABA-B receptors are working as 7-pass. That means it is passing like this, right? As you can see, these GABA receptors, they're passing like this. Now, this is a GI coupled receptors. That means it inhibits the cell from functioning. We'll go discuss the GABA receptor in the ligand-gated receptor. So GABA has two types of receptor. One is the GABA-A, another is the GABA-B. The GABA-B is a GI coupled receptor. That means it inhibits the intracellular functioning. Next thing is something from your book, that means the First Aid 2018, that in your endocrine chapter, you'll find this list that what are the several example of the receptors. Usually, if you know the mechanism of the receptor, that does not suffice your idea about these receptors, because even after you know about these receptors and how they work, you need to know that what are the examples or what are the ligands that are binding to this receptor, right? Now, as you see, that CAMP mediated pathway, these receptors or these hormones, this FSH, this LH, this SCTH, the TSH, CRH, HCG, ADH, these receptors and a lot of others, they work through the CAMP pathway. That means they stimulate the CAMP pathway or the GS coupled receptor. CGMP pathway, as you see that this receptor BNP, brain natriuretic peptide, atrial natriuretic peptide, endothelial derived, what is it here? Okay, nitric oxide, relaxation factor, endothelial derived relaxation factor, nitric oxide, they work through CGMP. We haven't discussed it yet, right? We discussed the CAMP mediated pathway. We discussed the protein kinase C mediated pathway, but we haven't discussed the CGMP. We'll discuss it. Next is the IP3 mediated pathway. As you can see, the gonadotropin releasing hormone, oxytocin, ADH, these are following the IP3. Then there are some receptors. Some of the receptors are intracellular receptor, as I told you earlier, that some of the molecules can cross the cell membrane and their receptors are inside the cell. So those receptors are intracellular receptor, because those molecules, this progesterone, estrogen, or as you can see, your thyroid hormone or vitamin D, they can cross the cell membrane and they can bind to a specific receptor inside the cell. So these molecules has intracellular receptor tyrosine kinase. This receptor has intrinsic tyrosine kinase activity like insulin and other growth factors, and non-receptor tyrosine kinase. These molecules, these prolactin immunomodulators, and these receptors have no intrinsic tyrosine kinase activity. They take help from other tyrosine kinase to do their job. Now, we discussed up to CMP, IP3 receptor. Let's see what else there. Now, ion channel receptors. As you see that here are several types of ion channels, like ligand-gated ion channel. This ligand-gated ion channel means whenever a ligand binds here, that is a neurotransmitter, is binding to that receptor, this ion channel is activated. Mechanically gated ion channel, due to some mechanical effect, like pressure on a cell, these ion channels are opened up. Right? Next is some ion channels, those are always open, like potassium leaky channels. We know that in the cell, there are potassium leaky channels through which potassium goes out of the cell. And here is the voltage gated channel. Voltage gated channel means due to the change in the voltage, let's say a cell is depolarized, then that cell will activate more other channel. One example, the myocardial cell. That when the myocardial cell are excited, then there will be opening of lots of sodium channels so that more sodium can come in and makes the other cells, this adjacent myocardial cell, to be excited or depolarized. Now, this thing is a voltage-gated ion channel. So these are the types of the ion channels. First, discuss the ligand-gated ion channel. Now, ligand-gated ion channel means that whenever a ligand binds to that ion channel is opened. Simple. Now, here you see that gated ion channel receptors open with signal binds or open when the signal binds. As you see, the ions are outside the ion channel. This is the ion channel and it is closed. But whenever your ligand that binds here, then as you can see, this ion channel is opened and ions are moving across. Now, that is your ligand-gated ion channel. Now, examples of ligand-gated receptors. What are the examples of ligand-gated receptors? Nicotinic cholinergic receptor. This is a ligand-gated ion channel receptor. What is this? Serotonin 5-HT3 hydroxytryptamine receptor. That is another ligand-gated receptor. And there is the glutaminergic receptor. That is another ion channel receptor. Now, about this glutamine, how this glutamine works, that is kind of interesting. That we know that glutamine has the excitatory function, right? This glutamine has excitatory function inside the cell. And as you see that it is a neurotransmitter, so this glutamine or glutamate, it is releasing from the nerve ending. This is a nerve ending. So if I draw the whole nerve here, you can understand that this is a neuron and this is a nerve ending, and this glutamine are getting released here. Now this glutamine, as you can see, it can bind to another nerve ending. That means in the synaptic junction, it binds with another nerve. And here, it stimulates these two receptors. One is the AMPA receptor, and there is the NMDA receptor. Now this AMPA receptor stimulation of this glutamine leads to increased sodium influx inside that nerve cell. And whenever cell gets positive charge, cell becomes depolarized and cell becomes active. Now, this way, this glutamate is activating this nerve cell. Now, also, you see that this glutamate has activity over this NMDA receptor. And whenever it binds to the NMDA receptor, two things happen. One, here's a small green ball. That ball is magnesium. Whenever this receptor binds, whenever this glutamate binds to this NMDA receptor, this magnesium thing is removed. Magnesium is removed from that channel, and this channel, whenever this magnesium is removed, you can guess that this pathway is now free, right? If I remove the magnesium from here, you can see that this path becomes free. And when this path becomes free, calcium goes in. And when calcium goes in, what happens? Another positive charge that leads to increased activity of the neurons, because positive charge leads to depolarization, and depolarization means increased activity. Very simple. That's how your glutaminergic neurons or glutamine neurotransmitter works. Now, we discussed about the GABA receptor, GABA B receptor, which is a G protein coupled receptor. Here is a GABA A receptor, which is not a G protein coupled receptor. It's a ligand gated ion channel receptor. And this thing is an ion channel for chloride ion. And as you see that this GABA this is a GABA receptor, GABA binding ion channel. And this ion channel has this several um, component like alpha, beta, gamma component. And GABA binds here, somewhere here and here. And when it binds here, it opens up the ion channel. So these 5 molecules are kind of clustered together. Now when the GABA binds here, it opens up. Now when it opens up, ion can pass through this channel. Now, the thing is that 2 drugs, one is the benzodiazepine and there is the barbiturates, right? The sedative medication. This benzodiazepine, it binds to a certain point in that GABA channel. These are agonists of this GABA channel. That means they increase the activity of the GABA. Now, how they increase the activity? Benzodiazepine increase the frequency of opening of the GABA channel. That means it binds to the GABA receptor and they opens up this ion channel more frequently. And this barbiturates, they does it by increasing the duration. That means they binds to this receptor, they opens this channel for a longer period. But both of the cases, there is increased chloride influx. And when the chloride is inside the cell becomes more negative charged. And when the cell is more negative, it means the cell is hyperpolarized. And these cells are not excitatory. And whenever we are not excitatory, what do? We go to sleep. Now, this cell will go to sleep because there is more negative charge inside the cell. That's how GABA A receptor works. Glycine, almost same way as GABA, they works as a ligand-gated channel. They does the influx of chloride and there is negativity inside the cell. That's how this ligand-gated ion channel works. Now, so far, we discussed the G protein coupled receptors. That means the 7 PAS receptors. And we discussed the ion channel receptors. Ion channel receptors, G protein receptors. Now, left is this 2, the receptor with intrinsic enzyme activity. That means receptor tyrosine kinase. And another is the non-receptor tyrosine kinase. Let's go over it. Now, here you see that enzyme link receptor. First is the receptor tyrosine kinase. Okay. Now this receptor tyrosine kinase, as you can see, that here these are the receptors, right? Now these receptors are wide apart, but whenever the ligand binds here, let's say these 2 ligands are binding here, these receptors becomes dimerized. That means they come close together. And when they come close together, they have some activity inside. This activity, this receptor, these are self-sufficient, or they are kind of self-employed because they have their own tyrosine kinase activity. Through that tyrosine kinase activity, what it does, they phosphorylate some tyrosine residues in their body. That means this receptor, if you can imagine that there are some tyrosine residues attached to them. Some tyrosine residues attached to them. Now, when this receptor binds with their ligands, they stimulate some enzyme intrinsic to them. Those enzymes are called tyrosine kinase. And whenever you see the word kinase, what comes to your mind? Kinase means some enzyme that adds phosphate to its substrate with the expenditure of ATP. Now, this tyrosine kinase will add phosphate to this tyrosine residues with the expenditure of ATP. Now, that's why you see this phosphate groups attached here. They're not showing very intricately the tyrosine residues, but you can guess that here is the tyrosine residues and where these phosphate are binding. All right. Now, next thing is that when these tyrosine groups are phosphorylated, they are activated, and that activated inside portion of that receptor makes some inactive protein to active protein, and there is certain action that runs afterwards. So that is the basic of the tyrosine kinase receptor. Now, what happens inside that you need to know? The first thing is you see that we know that insulin binds here in some tyrosine kinase type receptor. The several growth factors binds in this tyrosine kinase receptor. Now, as you see that these growth factors are binding to this receptor, and this inside that receptor, there is certain residues here, the tyrosine residues, and those are phosphorylated by the tyrosine kinase activity. After that, they bind to some protein. These proteins are sometimes called the relay protein. That means they will relay the action to some other protein. Do you see the relay race where the person, the athlete, runs and passes some kit to another person, then this person runs and passes this kit to another person, like this. And that's how it works. This SH2 domain is a protein that is a relay protein. It will bind to this phosphorylated tyrosine residues, and it will pass this downstream signaling. Now, what is the downstream signaling? As you see in this picture, the receptor tyrosine kinase, that means these ligands are binding here, the receptors are dimerized, the tyrosines are phosphorylated, and here you see one of the relay protein. Previously we discussed SH2. It can be different relay protein. In this picture, they're showing GRB2. That is another relay protein. Now, this relay protein binds with another protein. That protein is called SOS. And this SOS stands for son of 7 protein. I'm not sure why they're called son of 7. Now, this son of 7 protein, they're stimulated, and they stimulate another relay protein, the RAS protein. This RAS protein stimulate another protein that is called RAF protein, or simply we say RAS and RAF. And this RAF protein stimulates another enzyme, the MAP kinase enzyme. MAP kinase activating enzyme that stimulate the MAP kinase. And this MAP kinase, it will bind to some intrinsic transcription factor or stimulate that transcription factor. And that transcription factor stimulate the gene inside your nucleus to be activated, to be transcribed to some mRNA, so that that mRNA will produce some new protein. Again, this is a cascade of events that happens here. First, there will be the receptor ligand interaction. The ligand binds to the receptor dimerizes, then this receptor dimerization leads to stimulation of the tyrosine kinase. Then this tyrosine kinase phosphorylate the tyrosine residues along with that receptors, and this phosphorylated tyrosine kinase, tyrosine residues will bind to some relay protein like SH2 domain or GRB2 domain. There are several other domains, and when they binds there, they binds with the son of 7 protein. This whole group stimulates the RAS stimulates the RAF stimulates the MAP kinase activating protein, and this MAP kinase activating protein stimulates the MAP kinase, and this MAP kinase phosphorylates some transcription factor, and that transcription factor binds to your gene to transcribe some mRNA from that particular gene so that this gene can be transcribed and translated certainly or subsequently into some protein, and that protein will help in the growth. That's how your receptor tyrosine kinase works. Now, in the same picture, you see the non-receptor tyrosine kinase. Now, this non-receptor tyrosine kinase, as you see, that this has no intrinsic tyrosine kinase activity. It needs to take help from other enzyme to phosphorylate its tyrosine residue. Now, it is also a tyrosine kinase activity. That means here is also some tyrosine present here, and these phosphate groups are phosphorylated tyrosine. They're showing up here, but they have no intrinsic tyrosine kinase activity. This receptor takes help from other protein or other enzyme to phosphorylate those tyrosine. Now, these are the enzyme genus kinases that helps in phosphorylation of the tyrosine residues for this particular piece of receptor. Now, when these receptors, when these tyrosine residues are phosphorylated, they stimulate this another protein, the STAT protein. This STAT protein, now this whole STAT protein, as you can see, phosphorylated STAT protein, they run and they go inside the nucleus to stimulate some transcription factor to work as a transcription factor, and that stimulate the gene to be transcripted to mRNA, and from the mRNA, you know what happens? The protein is translated. Now that's how your JAK-STAT pathway works. Now, what is STAT? The full meaning of STAT is signal transducer and activator of transcription. Signal transducer and activator of transcription. So now it is transducing the signal. Not only that, this thing is also activating. This STAT is also activating the transcription. That's why the together is called signal transducer and activator of transcription. Now, what is next? The next is some other receptors. These are much more simpler discussion, like ANP receptor, atrial natriuretic peptide receptor. When we get ANP, when there is increased blood pressure, like there is increased blood volume, maybe due to heart failure, the blood cannot move on. There is stretching of the myocardium, and from the myocardial cell, there is release of this protein that is atrial natriuretic peptide, which go and leads to natriuresis. That means it leads to sodium urasis. Sodium goes out of the body, so that with sodium goes water, so water is lost from the body, and this stretching function or this increased stasis of the blood volume is kind of decreased. Now, how they works? They works through cGMP. They binds to some protein, some GProtein. This I'm not sure if it is. Let's see that atrial natriuretic peptide. If it is a GProtein coupled receptor. Okay, that is not shown here. They are just saying they're bind to the cGMP. I think this is a GProtein coupled receptor, because as you can see that GTP is working here. When it is a GProtein coupled receptor, it stimulates now this thing, this atrial natriuretic peptide and brain natriuretic peptide, that stimulates the production of cGMP. Now this cGMP stimulates protein kinase G, that's one thing. Another thing is they have some direct action. That means whatever the action is, like here in this ANP and BNP, what it does, they stimulates the natriuresis. That means they will release or they will excrete more sodium. How they can do it? They may put on some sodium channel so that sodium is more in the lumen of the nephron. Now, that's how this cGMP works. But this is cGMP. This thing we never discussed. Another way this cGMP is activated is by nitric oxide. So let's see how the nitric oxide and cGMP works. Now, before going to the nitric oxide, okay, nitric oxide is later. So we'll go to the nitric oxide later. Next thing is the intracellular receptor. The intracellular receptors are for the ligands who can pass through the cell membrane. Now, as you see, that here's extracellular fluid, here's the here's the cytoplasm. Now, these signaling molecules, which are small and which are lipophilic, they can pass through this cell membrane. And when they pass through the cell membrane, they can bind to this intracellular receptor. And when they bind to this intracellular receptor together, as you can see, this receptor and ligand, they can go and bind to the DNA to make some change, like make the DNA to be transcripted and to be translated. More detail here that cortisol. Cortisol has an intracellular receptor, how it works. Now, as you see that this cortisol is the signaling molecule and it is passing through the cell membrane, the plasma membrane, and this cortisol receptor is inside the cytoplasm, and this receptor has some protein bound to it. These proteins are called the chaperone protein, or sometimes we call it heat shock protein, or some protective protein. It is not always open. It's kind of a protective protein. And whenever this cortisol comes and bind here, this protective protein is removed. And whenever this protective protein is removed, this whole thing can now pass through this nuclear pore or nucleoplasm pore and go and bind to this DNA that leads to transcription and eventually translation. That's how your cortisol works. That means cortisol receptor is inside the cell, and this cortisol receptor is bound to some heat shock protein, the chaperone protein. Whenever the ligand binds here, that means cortisol binds here, the chaperone protein is removed, and this whole complex goes inside the nucleus, and they can bind to the DNA, and they can run the transcription and translation. Now, one thing is important to remember, that this receptor activity, or this receptor cortisol activity, we sometimes call it zinc finger receptor. Remember that these receptors are made up of zinc moieties, and whenever this, let's say this is the zinc finger receptor, and let's say this is the cortisol, all right? Now this cortisol binds here in this whole receptor complex, and what happens, this zinc finger kind of unfolds. Now when they unfold, they binds in the grooves of the DNA and works as transcription factor. Like if I draw this thing like this, that let's say this is the folded zinc finger receptors, okay? And here's the cortisol binding site. Now whenever cortisol binds here, this receptor unfolds like some finger, right? And this unfolding of this receptor, this unfolding, they will set in some DNA binding site. That means some grooves of the DNA. And when they bind to the grooves of the DNA, like here or here, let's say here, these grooves of the DNA binding can work as a transcription factor, and they start the transcription. That's how the zinc finger molecule works. Next thing is the nitric oxide receptor. Now, nitric oxide is a vasodilator. We all know it. Now, how it works, it leads to activation of cGMP. Let's see. In the endothelial cell, the nitric oxide is produced. In the endothelial cell, the nitric oxide is produced from some amino acid that is called arginine. From the arginine, this nitric oxide synthase works on this amino acid arginine to convert it into nitric oxide. And now this nitric oxide, how this nitric oxide synthase is working, some receptor mediated action. Maybe usually we know that M3 receptor, that means the muscarinic M3 receptor has a nitric oxide releasing activity. That means this receptor is the M3 muscarinic receptor, and this is a GQ coupled receptor that leads to activity of the calcium. We know how the GQ works by IP3 and DHE. I hope you remember. Now this calcium stimulates this nitric oxide synthase, and that converts the L-arginine to nitric oxide. And from this endothelial cell, this nitric oxide will be dissolved or will pass through this endothelial cell to the smooth muscle cell of the blood vessel, and where it will stimulate the guanylyl cyclase, and that guanylyl cyclase will convert the GTP into cGMP. So nitric oxide works through the guanylyl cyclase that converts the GTP into cGMP, and this cGMP leads to relaxation of that blood vessel. How it is relaxed? I hope you know that this smooth muscle relaxation is uh, done by the myosin light chain kinase phosphorylation. That means myosin, and myosin is a protein component of the muscle cell, and myosin light chain kinase phosphorylation. When this myosin light chain kinase, this enzyme is phosphorylated, it cannot work. And when it cannot work, there is no muscle contraction. And then there is muscle relaxation that leads to this dilation of the blood vessel. So again, how nitric oxide works? Nitric oxide stimulate the guanylyl cyclase that converts the GTP into C. All right. So the next thing, as you see, this whole functioning is shown in much more bigger picture. That acetylcholines are binding here. Maybe this is a M3 receptor, and that leads to, because M3 receptor is a GQ coupled receptor, so you'll find IP3, you'll find the release of calcium from the endoplasmic reticulum. There will be calcium binding calcium modulating protein, calcium calmodulin activity. This thing activates the nitric oxide synthase that converts the arginine into nitric oxide and citrulline. This nitric oxide go to the smooth muscle cell. That means from the endothelium, it goes to the smooth muscle cell and it stimulates the guanylyl cyclase that, okay, someone raised their hand. Any question? Okay, if you have any question, you can ask right now, no problem. I have two raised hands. All right, type down your question. Let me finish. Meanwhile, that this guanylyl cyclase converts the GTP into cyclic GMP, and this cyclic GMP activates the protein kinase G, and that leads to muscle relaxation or dilation of the blood vessel. Okay, so uh, that's how the relaxation or vasodilatory action of the nitric oxide works. So, what is your question? Uh, Write it down. There's a question answer bar, there's a chat box. You can write down your question here regarding this. Question? Okay, so you raise hands. That's why I thought you may have it. Okay, got it. No receptor or GQ, nitric oxide receptors are GQ or GS coupled receptor. Okay, that's a very important thing. How the nitric oxide works. Nitric oxide is a very specific, okay, nitric oxide, these receptors, actually, as you can see, that nitric oxide is a gas, right? Now, it directly works on the enzyme. It directly stimulates the enzyme, the guanylyl cyclase. It has no receptor in the cell surface. That means it will not bind to the cell surface receptor. It is going inside the cell and stimulating the enzyme, the guanylyl cyclase. It has no receptor. Okay, the nitric oxide directly go inside and binds and stimulates the guanylyl cyclase. No receptor. The receptor we are discussing here, this acetylcholine receptor, this is a muscarinic receptor. Again, okay, I think where's the confusion? Here you see that in this picture, let's say, on this chart, Where is the chart? Here. Here you see that one of the type of the muscarinic receptor is the M3. And this M3 receptor is a GQ coupled receptor. And this GQ coupled receptor, how it works by IP3 and DAG, right? Now that thing works on the exocrine glands and endothelium. And in the exocrine glands, it leads to secretion. In endothelium, it releases nitric oxide. Got it? So this receptor, which is the M3 receptor, the ligand for it is acetylcholine. The ligand for it is acetylcholine. All right. Now let's go to that discussion again. So now here you see that this is the blood vessel here, blood vessel. In the endothelium of that blood vessel, there is this M3 receptor, right? And acetylcholine will bind to this muscarinic receptor. This is a G-protein receptor, and particularly this is GQ coupled receptor. Now this GQ coupled receptor, when it binds with its ligand, there will be activity of IP3, and this IP3 activity will stimulate the release of calcium that binds with the calmodulin, that means calcium modulating protein. And together, this calcium calmodulin complex will stimulate the nitric oxide synthase. And this nitric oxide synthase will synthesize nitric oxide from arginine. This arginine is present in this endothelial cell. Now, when this arginine is synthesized or converted to nitric oxide, this nitric oxide now dissolves through the endothelial cell to the smooth muscle cell of that blood vessel. That means if I draw this blood vessel diameter, first there will be endothelial cell here, right? Now, below this or beneath this endothelial cell, there is a smooth muscle cell. Now, this nitric oxide passes from the endothelial cell to the smooth muscle cell, and where it stimulates this enzyme, guanylyl cyclase, and that converts the GTP to cyclic GMP. And this cyclic GMP activates some protein called protein kinase G. And this protein kinase G relaxes the smooth muscle. And when this smooth muscle is relaxed, the blood vessel has a chance to dilate. That's how your nitric oxide works. Now, if I go to the previous picture, you can see that they're showing that nitric oxide is getting produced from L-arginine, and this nitric oxide is dissolving from the endothelial cell to the smooth muscle cell and stimulating the guanylyl cyclase. Now, where is this nitric oxide coming from? If you look closely, there is a receptor here, and they are not showing which receptor for it, but from our next picture, I mean the picture that is on the next, we discussed that the acetylcholine binds to this receptor, and this is a M3 receptor, and this M3 receptor is particularly GQ coupled receptor that leads to increased calcium in the body by subsequent receptor signaling pathway. And this calcium binds with calmodulin and stimulates this nitric oxide synthase. That's how you get your nitric oxide. Now this guanylyl cyclase converts the GTP to cGMP, and this cGMP leads to relaxation of that smooth muscle. That's how this nitric oxide works. Okay, so I think that's the end of it. So now, do you have any question after this? Any other question? Any confusion? Well, okay, write down your question. Don't worry, you have time. Do you want to talk? Should I turn off your microphone? Receptor tyrosine kinase, what? What is about receptor tyrosine kinase? Want to talk? Okay. Let's make your microphone works. Okay, so just let me just go over the receptor tyrosine kinase again, then I'll come back to your question. Okay, so here is the receptor tyrosine kinase. As you see, why it is called the receptor tyrosine kinase. Why it is called the receptor tyrosine kinase. And this thing is called the receptor tyrosine kinase because this receptor has its own intrinsic tyrosine kinase activity. That means when the ligand binds to this receptor has its own intrinsic tyrosine kinase activity. Through that tyrosine kinase, it can phosphorylate the tyrosine residues attached to it. So this thing, if I look at this picture, okay, this is a good picture. Now, here you see that this is the receptor, right? When this ligand is binding here, this receptor dimerizes. That means they are come closer. Now, not only that, they have intrinsic tyrosine kinase activity here somewhere. Now, this tyrosine kinase will phosphorylate, certain tyrosine residues here. Now, when they phosphorylate the tyrosine residues here, this phosphorylated tyrosine residue then will bind to some relay protein, like a H2 domain that is shown here. Maybe it is some other relay protein, like GRB domain. And when they bind to these domains, they run some other action, like this GRB2 binds with son of 7 protein, that binds with the RAS protein, RAS binds with ref binds with MAP kinase activator protein, because this is called MAPKK. That means the protein that adds phosphate to the MAP kinase, or simply the protein that stimulates the MAP kinase. And eventually the MAP kinase is stimulated. That leads to the phosphorylation of the transcription factor. This line, this straight line here, it is showing the DNA. And this DNA is getting transcripted, and there is transcripted, the product of transcription, the mRNA. And from the mRNA, certainly there will be a certain translation that will lead to production of protein, and there will be growth because these are growth factors, right? That's how your okay, ANP receptor. Okay, I'll go over it now. That's how your tyrosine kinase receptor works. All right, for the ANP receptor, as I say, that this ANP receptor is a G protein coupled receptor, and this G protein coupled receptor, this is not shown actually very clearly. This G protein stimulates the CGMP production. That means this G protein, maybe they stimulates the guanylyl cyclase, because we know that when you get CGMP, it is from the action of the guanylyl cyclase. That means GTP converts into CGMP with the help of, I think GC means this guanylyl cyclase. They stimulate this GC to convert the GTP into guanylyl cyclase, and this guanylyl cyclase works on its action, like it can stimulate the PKG, the protein kinase G, or it can directly to some action. That means, what is the action of the ANP? It will lead to natriuresis. That means it will lead to loss of sodium. How it can lead to loss of sodium? Maybe it will stack the renal tubular cells with sodium, so that sodium is much more released into the lumen. And when it is into the lumen, the sodium is going out of through the urine. That leads to natriuresis. And with sodium goes water, so the water level or the volume of the body goes down. All right, so someone is raising their hand so frequently. Let's see. Okay, that's how the G ANP works. All right, so I'm allowing Dr. Tonima to talk. Okay, so you can talk now. I think your microphone is off, and I unmuted your microphone. Oh, I couldn't unmute your microphone. Let's see. Okay, I'm not sure how. I'm not sure how to unmute your. Oh, your
হ্যালো
microphone is. Yes, yes, I can hear.
হ্যাঁ ভাইয়া আসসালামু আলাইকুম শোনা যাচ্ছে আমি কি বাংলায় কোশ্চেন করতে পারবো
No, please speak in English.
ওকে ভাই আই হ্যাভ some confusion about protein kinase C so actually what is the function of directly protein kinase C I mean just it is I thought it was I mean it is contracting or it is dilating
Protein kinase C.
yeah
Where we discuss the protein kinase C.
I mean my protein kinase C is the GQ coupled receptor
Okay.
so I mean from the DAG protein kinase C come so
Oh,
um,
okay, all right.
yeah, actually, Bhaiya, where the GQ couple receptor is stimulated, there are two stem, right? DAG and IP3.
Mm-hmm.
But always there is no need to be stimulated of DAG or IP3 both, right?
Mm-hmm.
Like M3 like alpha 1 receptor acts via IP3, right?
Mm-hmm.
As but it doesn't act by DAG or it needs both stem to be stimulated to act alpha 1 as for
আচ্ছা
example
আপনি বাংলায় বলেন।
হ্যাঁ হ্যাঁ আচ্ছা ভাইয়া যেটা হচ্ছে প্রোটিন যেমন ধরেন আলফা 1 একটা রিসেপ্টর এটা তো হচ্ছে আমার জিকিউ কাপল রিসেপ্টর তো লাইক এটা যখন আমার কাজ করে আমরা তো জানি যে ভ্যাসো কনস্ট্রিকশন করে রাইট তো তাকে কি আমার আইপি3 ডিএজি দুটো স্টেমের মাধ্যমেই কাজ করতে হয় তাকে তো মূলত শুধু আইপি3 এর মাধ্যমে কাজ করতে হয় তাই না যেহেতু সে স্মুথ মাসেলকে কন্ট্রাকশন করে
ওকে।
এই জায়গায় একটা কনফিউশন আছে ভাইয়া মানে প্রোটিন কাইনেজ সি এর আসলে কাজটা কি আহ, আসলে ফাংশনটা কি প্রোটিন কাইনেজ সি এর
Okay, okay.
ভাই
So
আমি কি বুঝাতে পারছি
okay, so you are asking that if in this case the GU coupled receptor, there is protein kinase C, there is IP3, both of them. So what is the function of
জি জি
both, or what is the exclusive function of protein kinase C, right? That means
মানে ভাই দুটোকেই তো কাজ করতে হবে না লাইক আলফা আমি যদি উদাহরণ হিসেবে আলফা ওয়ানের কথাই বলি সে যেহেতু স্মুথ মাসেলকে কন্ট্রাক্ট করতেছে তো তার ক্ষেত্রে তো আমার প্রোটিন কাইনেজ সি কে লাগছে না শুধু কি সে
oh,
আইপি
okay,
থ্রি
okay, okay. I got it. I got it. No, the thing is, when you work in this way, you have both protein kinase C and
জি
IP3, right?
জি
Now,
জি
in case of IP3, you are getting calcium. And as you
হুম
see, that protein kinase C is kind of decreasing the calcium, right? That means
জি
it is
জি
increasing the calcium current. Now,
জি জি
maybe there's a balance here.
ও আচ্ছা আচ্ছা আচ্ছা
Because if
ও
just only the IP3 is working, maybe it is hypercontracting that
জি
smooth
জি
muscle.
ভাইয়া
But when
জি
there
ভাইয়া
is, I think you got it.
জি ভাই আমার এখানে কনফিউশনটা এটাই লাগতো যে প্রোটিন কাইনেজ তো ক্যালসিয়ামটা কমাচ্ছে তাহলে এবার কন্ট্রাক্ট করতেছে কিভাবে হ্যাঁ ভাই আরেকটা কোশ্চেন আছে
Okay,
আমি কি
okay,
বলবো
okay, okay, no problem.
আচ্ছা ভাই এম3 হচ্ছে জিকিউ কাপল রিসেপ্টর রাইট
Mhm.
এটা হচ্ছে কি যদিও ভাস্কুলার স্মুথ মাসেলকে ডাইলেটেড করে মেইনলি হচ্ছে আমার নাইট্রিক মানে ডাইরেক্টলি হচ্ছে কি নাইট্রিক অক্সাইড বাট এম3ও তো আমার ডাইলেটেড করে ওই একই ভাবেই তাই না ওই জিটিপির মাধ্যমে সাইক্লিক জিএমপির মাধ্যমে রাইট আপনি কি
If
এটাই দেখাইছেন ভাই আমি কি এটা মিস মানে
yes,
এম3টা হচ্ছে
yes,
সাইক্লিক জিএমপির মাধ্যমে তো কাজ করে রাইট
yes. M3 works through cyclic GMP, not sorry. M3 works through GQ.
ভাই আসলে কনফিউশনটা হয়ে যায় হচ্ছে এখানে এম3 জিকিউ কাপল রিসেপ্টর এখানে কোনো সমস্যা নেই বাট সে আবার এভাবে কাজ করে না লাইক ফসফোলাইপেজ সি এর মাধ্যমে
No, no, no.
আর ডিএসি
Look at this
এবার
picture. Look
জি
at
ভাইয়া সে হচ্ছে কি একটু ডিফারেন্টলি কাজ করে মানে গুয়ানাইলের সাইক্লেস ডিফারেন্টলি কাজ করে যদিও সে জিকিউ কাপল রিসেপ্টর ভাই এটা রাইট এটা একটু কনফিউশন লাগতো আর কি সবসময়
Okay, so let me explain again. That
জি
M3, this GQ coupled activity of the nitric oxide
জি
release. Now, in this case, the Okay, to all that one of our friends, she has a little bit of confusion, so I'm going over this discussion again. That when this, how this nitric oxide works and how this M3 works, that's two different things. Now, in your endothelial cell, there is M3. There is M3, that is muscarinic receptor. And acetylcholine, when it binds to the M3, it leads to action of the GQ coupled receptor because M3 is a GQ coupled receptor. Now, this GQ coupled receptor works as it is. That means it will work through the IP3 and DAG and this thing. But here, they are not showing the DAG part. Maybe that is not that significant here. Only the IP3, and that leads to release of calcium. Now, this calcium, we know that calcium is an enzyme activator. Whenever calcium is there, it activates the enzyme. Whenever calcium is there, it activates the enzyme. Now, this calcium activates the nitric oxide synthase, and then this nitric oxide synthase will stimulate the production of nitric oxide from arginine. And that nitric oxide will dissolve through the endothelium, comes to the smooth muscle cell, and does its function. It's not that the M3 is working through guanylyl cyclase. No. M3 is doing its job through the GQ coupled receptor or as the GQ coupled receptor works. But when it produces the nitric oxide, that nitric oxide stimulates the guanylyl cyclase to work on the GTP to convert it into cyclic GMP. And that cyclic GMP runs the other cascade of events. That means it stimulates the protein kinase G, and protein kinase G works on the muscle, other protein like myosin light chain kinases to relax the smooth muscle. Is it clear now?
Yeah, it's clear. Thank you.
Mm-hmm. Any other question?
No,
Okay.
thanks.
All right. Okay. All right. So that's the discussion about the receptors and intracellular signaling. I'm not sure how helpful it is, but let me know if you have any question after this. Okay, let's see. What was her question? Okay, what was her question? Her question was that first question was that how the IP3 and DAG works in the same place. That means in case of GQ coupled receptor, that how the IP3 and DAG works at the same place. Like here you see that IP3 is working, the DAG is working. Now both of them are working here. Yes, they balance out each other. They balance out each. That's what I said. Okay, I think you got it. That means that DAG is working on the protein kinase C, and it is decreasing the calcium. IP3 is increasing the calcium. Now, when both of them works, then it balances out each other. That means there is not hyperactivation of anything. All right. Next question was that why the how the nitric oxide works. Is it through any GQ coupled receptor or not? No, nitric oxide directly works in the guanylyl cyclase. But how we get nitric oxide, that is through the GQ coupled action of acetylcholine. This acetylcholine, when it binds to the M3 receptor, which is a GQ coupled receptor, which by the cascade of these events stimulates this nitric oxide synthase, and then we get nitric oxide. Then this nitric oxide has its own action with the guanylyl cyclase and GTP to cyclic GMP conversion, and eventually the muscle relaxation. Right? Okay, so any more question, any more confusion? Anything else? All right, so I think that's it for today's discussion. And it was great because this thing is kind of confusing, although sometimes we just memorize the how the receptor works. But if you know how they really works, I think that will help you memorize it much more quicker. Thanks for joining the session. I'll send you the email that contains this note, this lecture note. And no, all this pathway, I mean, not in intricate details you need to remember. You can remember these pathways, how they exactly works. That means if I say how the alpha 2 receptor works, you'll say GI coupled receptor. If I say, okay, how the M3 receptor works, you'll say GQ coupled receptor. Now you have to have this idea that what is happening here. That means in GS coupled, there is CAMP. In GQ coupled, there is IP3DAG. In GI coupled, there is decreasing CAMP. In case of nitric oxide, there is CGMP. This kind of idea you need to have in your mind. In insulin, there is tyrosine kinase activity. In case of cytokines, there is non-receptor tyrosine kinase activity. This type of idea, you will get it eventually. I mean, not at the first day. You can remember it. Whenever you read it, you review it, eventually it will be in your mind. So don't worry, don't need to memorize right now, but you will memorize it eventually. Trust me. So that's it. And take care, everyone. And I'll send you the notes. Have a good day. Goodbye.